Good morning, everyone, and thank you, Dr. Desh, and Siri, and the rest of the organizing committee for this event, for the invitation to be with you all today, and to share a little bit of our work and what I like to say are the applying research into the tiniest of tools at the nanoscale that we hope will hopefully make a bigger impact to our smaller patients kind of down the road. This is better. So, obligatory disclosures because making the lawyers happy. But before we kind of get into CF, like Dr. Desh said, I'm actually a pediatric hematology oncology physician, so I haven't had the privilege of taking care of kids with CF since my residency days. But as we kind of approached my training as a physician scientist and building our group, we've try to take a disease agnostic approach to the nanotechnologies and tools that we want to develop, and really cater those tools to the particular application. And so our group really thinks about it, and I kind of want to zoom out for a little bit before we get to some of the CF work in terms of thinking about precision medicine, where we want to basically tailor therapies to the individual patient. And so rather than, let's say, control the CF pathology, as we move towards gene therapies where we can move towards a pathway to a cure for CF, there's going to be different sets of tools and different needs that physicians and scientists are going to need to apply in order to make these medical interventions happen. And so our lab kind of thinks about this transition in terms of, tools and technologies that can basically develop the big data or harvest the big data that are required for these approaches. We're thinking also about applying what are called 3D bioprinting technologies to create basically model systems of the tissues that we're trying to target. But the focus of this talk is really going to be in our work in terms of manufacturing platforms for gene and cell therapies, which will hopefully allow us to ultimately deploy these types of therapeutics more broadly and hopefully democratize these therapies to more and more of our patients. So when we think about emerging successes in developing an engineered cellular or gene therapy, really the thread that connects these areas together, whether you're talking about a CF gene therapy or exciting developments in my clinical specialty in terms of immuno-oncology and developing and harnessing the immune system to attack a cancer, or in gene-modified stem cell therapies that are being applied for patients with hemophilia or sickle cell disease, a thread that really connects these areas together is that at some point, for a lot of cells, and in some cases hundreds of millions to billions of cells, you have to deliver some kind of package, some kind of parcel, a gene editing, CRISPR-Cas9 gene editing material, some kind of parcel to those cells quickly, efficiently, safely, and hopefully, cost effectively, because these therapies, unfortunately, right now are not cheap. Clinical grade gene therapies for let's say CAR T cell therapies in the immuno-oncology space cost about $500,000 a dose. When you think about the gene therapies that are coming out for different types of retinal disorders or sickle cell disease, hemophilia, you're probably approaching, not even including the medical costs, $1 million a dose. And that owes to a lot, because that really owes to the fact that to make these types of interventions, you really need very specialized equipment, facilities, and special types of what are called viral vectors that have really enabled this field. And so we think, and we'd like to see if we can do better. Because while viral vectors have really enabled these fields, and they have decent and reliable delivery, they've sort of evolved their nature's delivery service, if you will. They they have issues in terms of, cargo carrying capacity and, manufacturing costs in terms of creating kind of clinical grade, what are called good manufacturing practice grade viral vectors. Non-viral vector methods, similarly suffer from reliance on the need for complex instrumentation and in some cases scalability. And there really hasn't been an approach that kind of fits these ideal criteria to really, let's see, this is the laser, develop an approach that is both GMP compliant, is agnostic to cargo, can be delivered efficiently at high throughput with minimal toxicity, is scalable, can be sustainable if you're talking about ex vivo harvesting of cells. For CF, it's a different issue because you're trying to make an in vivo delivery and ultimately cost effective. And so our group really thinks about this problem in terms of a portfolio of different technologies, because we don't think there's going to be a silver bullet. And so in terms of delivery, we think about, let's see, laser, strategies for membrane permeabilization, as well as penetrating cellular membranes to deliver the cargoes using tiny nanostructures or transdermal-based approaches. And in the case of CF, which we'll talk about in just the next couple of slides, we're thinking about ways to nebulize the cargo in nanoparticle shipping containers that can reach the cells of the airway to achieve the gene therapy and correction of CFTR. And so that's really the crux of the issue, is that from our perspective, is that there are a lot of amazing colleagues of ours at UCLA and and on the Zoom call probably, where a lot of their research is focused on the gene therapy space in terms of developing the cargo. How are we going to achieve that durable correction to mitigate the pathology of CF? But what we think is missing is kind of the equivalent of like Amazon or FedEx, right? So, Amazon and FedEx became indispensable in your lives because they figured out how to put things in better boxes and get those boxes where they need to be. And so that's kind of a taste of what we're trying to achieve. And this is, it's just a concept that's not unfamiliar in the CF space. The concept of nebulized or aerosolized based inhalable gene therapies for CF is not new. But what has not yet really been achieved, and this is an old slide, and I know that there's really exciting work being done in delivering mRNA encoding CFTR cassettes that mRNA cargoes that encode CFTR for kind of transient expression and correction of CFTR in the airway. But there hasn't been an approach yet that has achieved this laser a single dose gene editing strategy for airway basal stem cells that need to be corrected in order to really correct the to develop a durable cure for cystic fibrosis. And so our team at UCLA is really taking a multidisciplinary approach to this problem, and we owe this really all to, and we're all really been brought together by my colleague Bridget Gompertz, who is, shout out to her talk. She's talking on Sunday, and she will have a lot more exciting stuff than me to talk about in this space, but she's really the person who's really brought us together. Together with our other colleague, Don Cohn, who is one of the pioneers of gene therapy at UCLA. He's developed different types of gene therapy approaches that have actually reached patients in terms of addressing diseases like severe combined different types of severe combined immune deficiencies, as well as sickle cell disease. So Bridget's group really provides us with a platform so that we can study and develop our delivery service. Don makes the specialized cargoes, and our group is really trying to figure out how to package those cargoes in a vehicle that can get to the right cells that Bridget's going to help identify for us. And so a little bit on the cargoes, when assembling a gene therapy, researchers need to really take into account, a variety of cargoes that enable gene therapies, and from different types of nucleic acids to gene editing tools like CRISPR-Cas9, or increasingly we're talking about new tools like base and prime editing machinery, and so it's sort of like I just drove up from LA, so you pack in your suitcase, you have to have your own little part for your shirts and your pants and whatever. And so we're trying to think about how to do that with these particles. But Don's group, as I said, is making the cargoes that we're going to use to hopefully develop a durable gene therapy. Paul Ayub, who is an amazing grad student in Don's lab, is really the leader of that effort. And really the goal here is to apply CRISPR-Cas9 based technologies, and this is essentially what our cargo is going to be for our particular box, to essentially make a cut at the CFTR gene, which then they can use to basically direct a corrected donor template to achieve what they call site-specific insertion of the CFTR gene. And an issue here is that these are big cargoes. You can't necessarily, if you have a small carry-on and you've got a surfboard, it's going to be, you're going to have some problems. And that's essentially where we kind of have an issue with, either current viral vector technologies or it's not a trivial task to do this with a nanoparticle either. And so part of what we try to do in the lab is try to establish ways to formulate nanoparticles to fit that surfboard into a more manageable package. I'm going to let Paul hopefully discuss with you guys next year if you guys invite him, and because he'll have his paper out and everything. But the cargoes that we're looking at make that cut at what's called intron 1 of CFTR, and he's designing different types of what are called guide RNAs to make that cut in an appropriate place. We use a technique called guide-seq to establish whether or not that cut is on target versus off target. And based off of that analysis, he has a set of guides that have minimal off target based off of his initial studies in what are called 16 HBE human bronchial epithelial cells. And so more on that from him to come. But the cargo is one problem, but there's also other barriers to accessing the airway stem cells. And in CF, I was just speaking just before coming up here with someone in the audience that you have to get through, if you're going to have a nebulized therapy, a thick layer of mucus, and you ultimately have to try and get to these purple cells, these airway basal stem cells, because those are kind of the long-lived progenitors that are going to lead to whose correction are going to lead to a lasting cure. That's a difficult problem because the airway also has tight junctions that kind of prevent things from getting there, getting to those cells. And so these are scanning electron microscopy images that one of our students took of a piece of airway tissue, and ultimately we want to get to the basal stem cells. How are we going to do that? And that's really kind of our job in terms of the nanoparticle packaging and how we approach that. So Ruth is a grad student in our group who has really taken on this challenge. We are kind of taking a 1-2 punch approach where we have one particle that can basically move in and disrupt the epithelium, and another particle that delivers the cargo. We wanted to have this all in one system initially, but I will get to that in a couple how why that might not be the best strategy. And so, in terms of our delivery example, what we were kind of how we're kind of describing this issue is that at some point you kind of need a snowplow to plow the roads in order to get the cargo where they need to be, where they need to go. And so it doesn't really work when you live in LA as an analogy, but sometimes I guess we can come up to where you guys are at. And so what are the different vehicles? There are different types of nanoparticle systems out there, from plasmonic particles and mesoporous and polymeric particles. Lipid nanoparticles have gotten a lot of contention. Our group has really focused on two types of particle systems in the past, in terms of using what are called supramolecular nanoparticles, which basically are these polymer dendrimer nets that basically can form cages around the cargoes by kind of like a Lego connection using what are called host guest chemistries. And we've applied those types of systems in proof of concept of gene therapy approaches with our colleague, Professor H.R. Sang at UCLA's Department of Molecular and Medical Pharmacology in proof of concept gene therapies for different types of retinal disorders, sickle cell disease, and Duchenne's muscular dystrophy. We've also kind of started to think about lipid nanoparticle-based approaches because the COVID-19 vaccines have really offered that platform as a really awesome clinically translatable solution for this work. We need to be able to test these particles in a way, and so Bridget's group has developed an expertise in developing what are called airway liquid interface culture models of the CF airway, where they can basically create a little mini airway that's basically floating on a cell culture insert, where we can basically then take our nanoparticles and use a nebulizer and nebulize straight onto the dish, which helps us kind of test these the delivery a little bit easier. So we have kind of the tools and the platform. So what makes the appropriate choice for what nanoparticle to use? And so when we compared initially the supramolecular system to the lipid nanoparticles, we went essentially with the lipid nanoparticle system because the supramolecular nanoparticles in this slide, this is an older slide, but it shows that some of the cells are kind of lifting off and don't look as happy. And so that caused us to really shift a little bit towards the lipid nanoparticles. We think, we might actually revisit this work because we've been able to tweak the SNMPs a little bit more to maybe minimize some of that effect. But for now, it's really charging ahead with these approaches for the lipid nanoparticles. And they're appealing because you can make them scalably using what are called microfluidic mixing approaches, which we'll get into in a little bit. They're modular by design, so we can kind of configure them for different applications, such as our need to make a snowplow versus a delivery truck. And there's clinical precedence to using them that you're in terms of the recent what we've learned from the recent pandemic. And so when designing lipid nanoparticles, there's really four components that you really have to think about. There's an ionizable lipid, which impacts the encapsulation of the cargoes that you're trying to deliver. A helper lipid that helps in something called endosomal escape, which is what we'll talk about in the next slide, and helps reduce some of the cytotoxicity. There's sterile components that help tune the rigidity of the lipid membrane, and ultimately a pegylated lipid that helps really prevent these particles from sticking together and kind of has other advantages that you want to help tune. So when a design consideration for these particles is that as the particles are actually basically kind of taken up into what's called an endosome when they reach a cell, and that endosome is basically a lipid jacket that the particles kind of swim through. And as the endosome matures, there's a pH shift, which you basically try to leverage to allow the part to engineer your particles to release as that pH shift is happening, to release your cargo. And then, depending on the type of cargo that you're trying to deliver, you might need to get to the nuclear envelope, or in the case of mRNAs, the cytosol is just fine. And so we take two different approaches to making these particles. We use microfluidic-based approaches because they're more scalable or a little bit more precise. Ruth, for her bespoke stuff, likes to do a little bit of this by hand when she needs to do something quick. But the idea is that there's a number of ways that we can do this to make these particles quickly in order to do the testing that we need. In terms of microfluidic approaches, you're basically trying to create areas where the components of the particles mix in such a way that they can self-assemble into the particle, so that the particle self-assembles. We've focused a little bit more on what are called chaotic mixing microfluidics devices, but we're recently moving towards basically what are kind of toroidal bifurcating mixers, which are kind of more the commercially preferred in the kind of the more common commercially available systems that are out there. When we apply these approaches to make the particles, what we see, and again using the 16 HBE cells as sort of as a model for this, when we load the particles with a mRNA cargo encoding for something called green fluorescent protein, which basically makes the cell turn green as a kind of a way to kind of visualize that the delivery is successful. What we try to do is really optimize the formulation. A lot of the work is really trying to optimize the formulation of the particles and figuring out what are the best lipid components that are going to lead to the maximum delivery. And so in initial work, we identified that this ionizable lipid, which is, as I said before, the component that really helps with the complex in the cargo, yielded, our best delivery results in this initial screen, the C12-200 lipid. But the vendor that we were getting it from was giving us a little bit of kind of it was hard, it was difficult to get to get quality product. So we expanded the screen and actually found that in a broader screen that the SM102, which is actually the lipid that's used in the Moderna vaccine, has given us, comparable performance in terms of delivery of the GFP mRNA, as well without sacrificing, the viability of the cells that we're trying to target. Another thing that we're trying to do, especially as we move towards aerosolization of the lipid nanoparticles, is to screen for the other types of lipid components, the sterol and particularly the PEG lipid type. And this is not our work. This is just a shout out to Goraya Saya's group at Oregon State, who did some beautiful work recently. It was published in ACS Nano that shows that there's an interplay between that PEG lipid and the sterol that you can use to really fine tune the stability of the nanoparticles for aerosolized delivery. And they were able to demonstrate that they could deliver their LNPs to the lungs of mice in a sustainable and a repeatable way. And so we kind of take inspiration for that as we kind of move forward with our work. We're also really kind of as we kind of apply those principles in our nanoparticles, we also see that the changing the sterol component can have an effect on the delivery performance. And what we're also seeing is that the type of lipid that actually anchors that PEG group also can have a substantial impact on the delivery that we see. And so these are kind of the lessons that we're learning as we're trying to design the different type of particles that we're trying to develop. And so in terms of that first particle, the snowplow, if you will, the idea is really to incorporate into the lipid nanoparticle structure kind of soapy molecules that are called basically surfactants that can help the particles penetrate into the mucus, but also create a transient injury or to temporarily disrupt the epithelial barrier to expose the airway stem cells that we need to deliver to. the surfactant that we've chosen for this work primarily is something called polydocanol, which is, actually a small molecule that's used by a lot of airway biologists to slough off the airway for in vivo testing in mice models. And it's a molecule that we kind of stumbled on by just basically kind of initial conversations with the Gompertz group. And Emily and Ruth are really focusing on developing this system in particular. And what we see is that we can, what's convenient about this molecule also is that it has a tail that we can use to help complex to the lipid structure itself. And so we find that we can incorporate up to about 40% of the polydocanol into the nanoparticle itself using a combination of zeta potential and dynamic light scattering based approaches and without really affecting the size of the particles and the consistency of the particles too much. And when we basically incubate these polydocanol doped nanoparticles and compare them to incubating the cells with straight polydocanol or no treatment at all. These confocal images, the blue is a DAPI, which is a nuclear dye. The red is keratin 5, which is used as sort of a marker of the stem cells. So in this case, without any treatment, the stem cells are kind of covered by a lot of these, a layer of cells on top. And as we introduce the surfactant or the surfactant complex nanoparticle, those stem cells get exposed as that layer kind of sloughs off. But after 24 hours, what we see is that injury is still more significant and the airway hasn't started to repair. Whereas in the case of the lipid nanoparticles, the hypothesis from this data is that we're basically exposing smaller pockets with the nanoparticle to the polydocanol, which allows us to would hopefully allow us to have a more transient injury so that we don't adversely affect things kind of down the line. We're looking at some alternative surfactants like lysophosphatidyl as well, which is a small molecule, a native small molecule in the airway that can that we're hoping to use for basically temporarily, transiently permeabilizing some of the tight junctions in this space. I said earlier that we tried to put this all into one nanoparticle at first, and this is really the data that really speaks to that. Where when we found that when we try and do everything all at once, our delivery performance kind of tanks to a certain extent, and so we figure we have to be kind of a little bit more like Muhammad Ali and kind of like have a 1-2 punch in that way. I'm showing this is actually data from this week, and we haven't vetted it as much, but I am only showing this slide to shoot out to the community, whoever is on this call or anybody here who does these tier measurements, because we'd really love advice on how to really optimize our protocols for this. But this is a technique that we're hoping to apply to really monitor the integrity of the epithelium long term, or in real time over a longer period over a longer observation period, so that we can hopefully get a little bit of finer understanding of the repair processes that might be going on with some of these epithelial disrupting particles. The next thing is really the delivery truck. And just to kind of in the last couple of slides go through that, we want to configure our particles for delivering cast iron cargoes. And so they're big cargoes, as I mentioned before. We want to also be flexible because Don's group actually prefers to use what are called ribonuclear protein complexes as opposed to mRNA. We've kind of fought for the mRNA because it's more we know that it's clinically translatable and they're smaller, so it's easier for us to pack. But we had an issue at UCLA, and I shouldn't even be saying this, but UCLA had an issue with the company that makes the mRNA for us. And so all the labs who were trying to get this type of RNA at UCLA couldn't get this stuff because someone wasn't paying the bills. And so we had to switch to the Cas9 RNP for a little bit, and we had to optimize our protocols for that. The good news is that it looks like we can actually get cutting of at a locus with these Cas9 products using a modified 16 HBE line, which we modified to express blue fluorescent protein. And the cool trick with this is that if you make a 3 nucleotide edit in blue fluorescent protein, you can turn it green. So now you have basically a stoplight based approach to either tell if you're doing gene editing or gene cutting. We're focusing on the gene cutting, so we're basically turning off the blue. And what we see with either our RNP or mRNA cargoes is that we can achieve, between hopefully a respectable 12 to 15% cutting at the BFP locus. When we switch to CFTR cargoes, we still have a little bit more optimization to do, but this is really the first step towards achieving that editing and delivering the CFTR editing machinery that we want to deliver. Ultimately, we need to aerosolize this. This is our kind of test platform using a nebulizer that actually is a acoustic vibrating mesh that basically suspends the lipid nanoparticles in these aerosolized droplets. We're trying to optimize the parameters for that aerosolization. And what we're finding in some of these initial studies is that the particles are basically pushed into that aerosol droplet through essentially a vibrating sieve or a mesh. And as they're kind of going through and informing that droplet, I think they're kind of coming together and aggregating a little bit. And so we're going to need to optimize we're working towards optimizing our formulations to kind of prevent that aggregation and hopefully have more consistent delivery. This is a pain to do all at once, one at a time, because it's just one little nebulizer and you have somebody kind of doing like this. So we're engineers, we have a 3D printer. We're trying to actually multiplex this so that we can do multiple combinatorial studies at the same time. And so Stefan, who recently found out he was accepted at UCLA grad school for his PhD. He was a technician in our lab. He's our 3D printing guru, is developing a platform for doing multiplexed screening of different LNP nebulized systems. And hopefully we can, apply this to other types of nebulized delivery in airway models in the future. That's kind of whirlwind tour. Hopefully I didn't talk too long. Looks like it didn't. But of what we kind of think about in terms of designing the packaging that's going to be required for cystic fibrosis gene therapies in the future. There's a lot that goes into designing the cargo, figuring out what the properties of that box that are going to be important for your delivery conditions and ultimately getting the particles where they need to be. And it's early stages, but hopefully this is a contribution to get to the real gene therapies that we're all looking for in this space and in this community. So these are the people that do all the work. I just get to be the quarterback or the Mandalorian, I guess, in this case. And we really appreciate CFRI and all of our funding support that have enabled this work and others. The CFRI grant has really enabled us to recently receive a CIRM award, which for folks in California is the California Institute of Regenerative Medicine, which is the state stem cell agency. And so our group team was recently awarded an award to kind of continue this work. And so thank you guys for enabling and seeding that this project to hopefully lead to bigger and better things. We're also expanding, so any students out there who are out there who want to play with some nanoparticles and 3D printers and stuff like that, we're looking, please reach out. Thank you so much for your time and attention, and happy to answer any questions.